Hi, today we are going to talk about translation. That's not the kind of translation as in from a foreign language into another language, but it's actually translating the code of RNA into a protein. So before we get to that, let's review very quickly. We know that all, at least eukaryotic cells, contain a nucleus where we have chromosomes, and that chromosomes contain DNA. And remember from just a few lessons ago, we learned that DNA is made of this sugar phosphate backbone, two of them. It's a double-stranded helix, and we have these base pairs that form the rungs. And we also learned about how DNA replicates itself and also how DNA uses transcription to create RNA. Here is just a very simple diagram showing a eukaryotic cell nucleus where the DNA is going from a DNA structure, using transcription to create some RNA, and then translation is going to be the next process that we are going to talk about, and that is where the messenger RNA is going to create a protein. So, translation is the formation of a protein from the messenger RNA. And there are a few more molecules involved besides just the mRNA. We're going to talk very briefly about them. However, this can be very complicated, and if you'd like to try for some advanced proficiency, you are welcome to research this more thoroughly because there are a lot of puzzle pieces to this. First, we have our messenger RNA. We start with that. And a ribosome from the cell in the cytoplasm is going to be attaching itself to that mRNA strand. Then we have other kinds of RNA, the tRNA. And here this is just portrayed as this shape here, the tRNA. And it attaches itself to the messenger RNA. And the messenger RNA has a very special code that it gives, that it sends out. And this tRNA reads that code and is able to add an amino acid, the appropriate amino acid that the code is for, to this string of amino acids, and eventually that string of amino acids becomes a kind of pre-protein, a string that's going to become a protein. So translation eventually stops when a code that says stop is reached. It's called a codon. And we're going to talk more about that in the next slide. Now you're probably asking, what in the world is a codon? What a strange word. And is it a special code? That's a really good question. If we look at this diagram here, we have right along here the backbone of an mRNA molecule, and it is divided into, the bases are divided into threes. We have a triplet here, and a triplet here, and a triplet here. And that is, each of those is a codon. A codon is a nucleotide triplet, three of them together. Three of them together, three of them together. Each one of them specifies for a specific amino acid or for a start or a stop signal. Start meaning we're going to begin production of a new protein, or stop meaning this is the very last amino acid, we're not going to do any more. So that's what those mean. This code GCC is specific for the amino acid alanine. This AUG codon is specific for methionine, which actually this is an incorrect diagram because methionine is a start code. And you'll learn how to read tables to know how to read these codes. These tRNA molecules here are represented, and they have an anticodon right here, which pair up with the mRNA codon. And these are very specific to give us just the right amino acid. Here they are showing amino acids carried by the tRNA, and they are joined to the chain of amino acids by these bonds here, peptide bonds. And so that is how the protein gets formed. Now, don't be afraid of this scary looking table. It might look intimidating, but really it's not at all. It is a table to help you read the codons and know which amino acids they are coding for. So here we have our 4 different possible bases for the first base. Across the top are the bases we could choose from for the second base, and along this side for the third base. So let's say we have on the mRNA chain, we have 3 bases, C and then a U and a U. That gives us the amino acid leucine right there. And you'll notice that some amino acids can be formed using not just one particular triplet, but there could be several different triplets here. There are 4 different nucleotide triplets that produce leucine. Here we have some different stop codons that produce those. Here was our methionine start codon, and these are the 20 different amino acids that get produced by these triplet nucleotide bases, and you need to basically know how to read this table. You certainly do not have to memorize it. So that brings us to a very important concept, and that is that this process is universal to life on Earth. All life uses proteins, and the way they are made is through translation. And just a quick recap, we have our DNA molecule here, and this is just a little diagram showing how DNA will go through transcription to form mRNA. And here you see how the codon is these 3 nucleotides together. That codon codes for a particular amino acid, and these code for this amino acid. Each one codes for a very specific amino acid. These are just abbreviations. And those amino acids strung together in a particular order will make a protein. We do have a few more things that need to happen to this string of amino acids before it becomes a full-fledged protein, and we're going to find out about that in the next chapter. But I tried to represent a lot of different kinds of life on Earth. All of these forms of life use this process. It is critical to everything that lives. Now, some ideas for advanced proficiency. If you're interested into looking into more of these details about how the tRNA molecules enable the translation process, that's a great research topic, or what the role of rRNA is. What are some of the differences between the prokaryotic and eukaryotic processes for translation? You can research different proteins. What do they do? What are they important for in the body, in cell function, in a plant, and whatever it is you're researching that. You can research something else that you have a great idea for, some questions that you had during this presentation. That would be really good. So thanks a lot, and we'll see you in class.